So we begin with today's module, that is embryology module. We begin with our code master MRCOG with concept oriented clarity. So today's topic is the gametogenesis, that is the formation of the male gamete as well as female gamete. The process of formation of the male gamete is called as spermatogenesis. The process of formation of the female gamete is called as oogenesis. Okay, we know well that the sperms, which are the mature cells, they are derived from a basic cell which is called as spermatogonia. Okay, so these spermatogonia are present in the seminiferous tubules of the testis and they are diploid in nature. Okay, so these spermatogonia, they undergo the process of mitosis and they will form the additional spermatogonia. Okay, so these spermatogonia, these are uh, capable of dividing and forming additional spermatogonia. Okay, so actually there are two types of spermatogonia type A and type B. So these type A divide and form additional spermatogonia, but once they reach to a certain stage, they become a spermatogonia type B. Okay, when there will be no further division, but they will get converted into the primary spermatocyte. And once they are converted into the primary spermatocytes, they are destined to form the sperm cells. Okay, so these primary spermatocytes, which are diploid in nature, they are formed from the spermatogonia. Okay, so they are diploid in nature. Here, the first meiotic division will start. Okay, because these are the gametes. Okay, so we know that it has to become a haploid cell. So the first meiotic division will form and it will be a haploid. Okay, so it will have 23 X chromosomes and you will get a secondary oocytes. Okay, from secondary oocyte there will be further second meiotic division. This is similar to mitosis where there is no reduction in the number of chromosomes, but there will be further it will also be haploid in nature and it will form the spermatids. These spermatids will then get converted into the mature sperm. Okay, this is the mature sperm which has got the head, the midpiece and the tail. Okay, we'll see further subsequently in the next slide. Okay, so similarly, the in oogonia, uh, this oogonia which are there in the ovaries, they will they are diploid in nature. Okay, so this oogenesis doesn't take completely in at a one stage. Okay, it happens partly in the fetal life, then partly it happens at the time of puberty, and later on further stages, few stages does happen after the fertilization also. Okay, so that also we are going to see. So basic cell here is a oogonia. This oogonia will give rise to only single ovum. While here, if you see in the previous slide, this from the oogonia, you are going to get only one matured ovum. Okay, so these oogonia, they will undergo initial mitosis, and after that they will undergo the first meiotic division. There will be arrest at the prophase 1. Okay, M1 of prophase. Okay, in the primary oocyte. Okay, so later on these primary oocytes will undergo second meiotic division. Okay, will undergo further complete this first meiotic division and the first polar body will be formed and it will become a secondary oocyte. And this secondary oocyte will further undergo another meiotic, that is the second meiotic division. Second polar body will be formed and you will get a mature ovum. Okay, but this will take place only when there is a fertilization. So second meiotic division will take gets completed when there is a fertilization. Okay, now this spermatogonia, we have seen how it gets from sperms are formed from the spermatogonia to the mature sperm. Okay, so gametogenesis is formation of the gametes. The gametes will develop into the gonads. So in females it is called as oogenesis, that is the formation of ova. In males it is called as spermatogenesis, that is the formation of the sperm. Processed by spermatogonia, which differentiate into spermatozoa, which begins at puberty. Okay, uh, spermatozoa are formed in the walls of the seminiferous tubules of the testes. So this is the process of spermatogenesis, and there are which are the cells which are involved in the spermatogenesis. One, these cells which are in the different stages of the development. Okay, this primordial germ cell in form of spermatogonia, then primary spermatocyte, secondary. These are the different stages of the cell stages in the spermatogenesis. The another cell is a Sertoli cell which is there. Okay, also called a sustenicular cell, and these Sertoli cells will provide the support and nutrition to the germ cells. Okay, this is regulated by the luteinized. This is like Sertoli cells are under the influence of the FSH, while the Leydig cells, they are under the influence of the LH. Okay, and these Leydig cells will stimulate the testosterone production. Okay, so they are asking these FSH and LH and Sertoli cells and the Leydig cell function. Okay, so Sertoli cells will provide the support and nutrition to the germ cells. These LH will act on the Leydig cells and will stimulate the testosterone production. They are also asking about the total duration. Okay, so total duration is 64 to 74 days. Few books mention 74 to 90 days. Okay, so it is around 3 months, you can remember. Then it requires lower body temperature, 2 to 4 degree below the body temperature. There is a blood testis barrier which is formed by the Sertoli cells. Okay, sperm become functionally mature in the epididymis. So this is also very important. From this information is important from the SBA point of view. Total duration, the requirement of the lower body temperature, that is 2 to 4 degree below the body temperature. The blood testis barrier is formed by the Sertoli cells. Sperms become functionally mature in the epididymis. Then Sertoli cells provides support and nutrition, and FSH is acting on them, while the Leydig cells are under the influence of the LH and they produce the testosterone. This all information is important from the examination point of view. Okay, so the same what we have discussed is shown in this figure. Okay, then this spermatocytogenesis, the spermatogonia, as I mentioned, there are initially they are type A type of spermatogonia, then they become intermediate, and then they form the type B type of spermatogonia, which will form the further sperms. Okay, so initial there is a mitotic division. Okay, but after that the meiosis will start and there will be reduction in the number of chromosomes. Okay, so from primary spermatocytes, which is 46XY, there will be first meiotic division. They will form the secondary oocyte, and 23X or 23Y will get formed. This secondary spermatocyte will undergo meiosis 2 and the spermatids will form. Okay, so there is a halving of the chromosome number. Clinical relevance is that if there is any error, it will lead to the aneuploidy or the infertility. Okay, so they are asking these basic concepts. Then next is a spermiogenesis. Okay, up till now what we have seen is a spermatogenesis, but next is a spermiogenesis. Okay, spermiogenesis is the maturation phase. Okay, the process of transformation of a circular spermatid to spermatozoa is called as spermiogenesis, also called as spermiogenesis. Okay, these spermatids will get converted into mature spermatozoa. What are the major changes which are happening? There is a nuclear condensation, acrosome formation, flagellum development, mitochondrial sheath in the midpiece, and the excess cytoplasm gets shed when there is a conversion from the spermatid to the mature spermatozoa. Okay, there is no cell division here. Okay, so from one spermatid to spermatid, you are going to get one spermatozoa. There is no cell division. Okay, and there is it is under the hormonal control, that is the testosterone via the Leydig cell. Okay, next is a spermiation. Okay, what is spermiation? The release of spermatozoa from Sertoli cells into the tubal lumen. The sperm are still immotile. Okay, they will get motility later in the epididymis. So this is the one thing which happens in the epididymis. Thus, there is a maturation of the sperms and they will become motile. Okay, they are asking these image related questions. So here you can have a image based question. They may ask you that they will label a particular part as X or Y, and they will ask you to identify what is X or what is Y. Okay, so here there is a head. Okay, this is the nucleus, and this is the vacuole, which or vesicle, which contains enzymes to dissolve its way into the eggshell. Okay, this is the nucleus which contains the chromosome. Then there is a midpiece which contains the mitochondria to release the energy for swimming, and this is the tail. Okay, so even the content of the midpiece you should know. It contains the mitochondria to release the energy for swimming. Okay, and actively motile, this spermatozoa is an actively motile freely swimming cell. It consists of head, neck, and tail. Head is ovoid in shape, consists of nucleus. The anterior two-thirds of the nucleus is covered by the acrosomal cap. Tail has got three segments, that is midpiece, principal piece, and the end piece. Okay, so these are the parts of the tail, that is the midpiece, principal piece, and the end piece. Okay, Now coming to the oogenesis. Oogenesis is also similar to the spermatogenesis. However, there are little differences. Okay, it occurs in the ovaries and it doesn't get completed in the adult life. It begins actually in the fetal life. So there are another very peculiar thing about oogenesis. There is a very finite number of oocytes and there is no postnatal renewal. So whatever the number of sets of oogonia which are present, they are present at the time of birth. Okay, there is no further division. So what happens in the fetal life? There is a proliferation phase. There are initially primordial germ cells. These primordial germ cells will migrate to the ovaries and they will differentiate into the oogonia. These oogonia will undergo process of mitosis. The peak number of the oogonia is at 20 weeks of gestation, which is around 6 to 7 million. This figure also you should remember. So peak oogonia happens at around 20 weeks of gestation and it is around 6 to 7 million. Okay, so the initial part is in the fetal life where this primordial germ cells will get migrated to the ovaries and they will get differentiated into the oogonia. These oogonia will undergo mitosis. The peak number is at 20 weeks of gestation, around 6 to 7 million. Okay, so the meiosis begins in the fetal life itself. Okay, so the oogonia will form the primary oocytes. The oogonia will form the primary oocyte. Okay, so there is entry into the meiosis 1. But what happens? There is an arrest at prophase 1 in the dictotiny stage. Okay, so this you should remember. So in the fetal life, these oogonia form the primary oocyte. Okay, and these primary oocytes will undergo meiosis, but meiosis has got a further substages. So in the prophase 1 dictotiny stage, this meiosis, there will be arrest and this primary oocyte is surrounded by the single layer of granular cells called as primordial follicles. So such primordial follicles are present in the ovary till the time of puberty. Okay, so this is what is happening in the fetal life. Then what happens from childhood to the puberty? There are no further meiosis. There is a progressive atresia of the oocytes. So at birth, out of 6 to 7 million which were present at around 20 weeks of gestation, there will be only 1 to 2 million oocytes. But there is further atresia and at puberty you are going to get around 3 lakh to 4 lakh oocytes will be present. Okay, which will be utilized throughout the reproductive career. Okay, so from puberty to the reproductive life, there is a follicular maturation under the influence of the FSH. This primordial follicle will become primary follicle, secondary follicle, which will get formed into the graphene follicle. Okay, that is just before ovulation. The primary oocyte will complete the meiosis 1. Okay, so what we have seen, this primary oocyte started its meiosis, but it has got a arrest in the fetal life into the prophase 1 dictotiny stage. Okay, so there will be further completion of the meiosis 1. Okay, at the time of puberty, and it will form the first polar body, and this it will get converted into the secondary oocyte. Okay, so secondary oocyte and the first polar body will form. Okay, then there will be ovulation. The secondary oocyte will be released and this is arrested at metaphase 2. Okay, so this secondary oocyte is arrested at metaphase 2. This is surrounded by the zona pellucida and the corona radiata. Okay, and what happens here is the first polar body and then you get a second polar body. This first polar body may divide or it may not divide also. The what is the fate of this polar body? They degenerate. Okay, so from one primary oocyte you are going to get only one ovum. Okay, so there is a first meiotic division, there is a second meiotic division, and from one primary oocyte you are going to get one ovum. But this does not happen at a stretch. The part of this happens in the fetal life. The part of it happens at the from the puberty onwards. Okay, but the meiosis 2 will get completed only after the fertilization. Though the meiosis 2 has started, okay, this secondary oocyte will undergo meiosis 2 and will form the ootid. Okay, and this second polar body at when it will get released when there is a fertilization. So if the fertilization happens, it triggers the completion of the meiosis 2. It will form the ovum and second polar body, and the male and female pronuclei will fuse and will form the zygote. Okay. So you should remember meiosis is arrested twice. One is in the prophase 1 in the fetal life, and second is in the metaphase 2 until the fertilization. Okay. So these are the two phases where there is a arrest in the meiosis. Okay. Only 400 to 500 oocytes are ovulated in the lifetime. Remaining follicles will undergo atresia, and increased maternal age increases the risk of aneuploidy. Okay. So this is what is the thing which you should remember here. Okay. So the initial part is in the fetal life. Okay. And further steps happen in the uh, puberty onwards, and this meiosis 2 will get completed if there is a fertilization. Okay. The same thing is mentioned over here, what we have discussed. Okay. Finishes the gametogenesis. Thank you.